Now I'd like to introduce our final speaker of today, Dr. Haoran Zhao is also with the Indoor Environments Group at Lawrence Berkeley National Labs. He is a postdoctoral researcher in Residential Building Systems Group and his work focuses on the fate, transport, and dynamics of novel indoor air pollutants and their association with building characters. His current work includes the laboratory and field experiments on evaluating mechanical ventilation effects on indoor air quality in buildings. Dr. Zhao?
That's for introduction. Hi, everyone. My name is Haoran. I'm a postdoc from Lawrence Berkeley Lab. And today I will introduce our laboratory studies on the airflow influence on the airborne contaminants indoors, which has been mentioned by Jacob in the previous presentation. So our topic is to investigate the influence of overhead HVAC on the exposure to airborne contaminants from simulated speaking in a meeting and a classroom. So this study was conducted by the colleagues from Sustainable Energy and the Environmental System Division at Lawrence Berkeley Lab, leading by Brett Singer as their PI. Next slide, please. First of all, I would like to acknowledge that this research was supported by the US DOE through the National Virtual Biotechnology Laboratory, and the use of FlexLab in this work was supported by the Building Technology Office within the DOE Office of Energy Efficiency and Renewable Energy. Next, please. So the objective of this laboratory work is to study mixing conditions with cooled, neutral, and heated supply air with overhead HVAC supply system in a typical meeting room and classroom. And the second objective is to investigate the effect of imperfect mixing on human expelled contaminant dispersion in fully sized room with optional occupancy. And the third is to investigate the potential impacts of stratification of exposure on airborne contaminants from simulated speaking when heating with overhead diffusers. Next, please. So we have two setups in our experiment. The first setup is a simulated physical distance conference room. So our experiment was conducted in FlexLab, which is an integrated building technology testbed that we can configure and instrumented to study how the overhead HVAC system on the airflows and the contaminants. So we set up this experiment in FlexLab, one of the cell of FlexLab, which is a 30 by 20 by 9 feet room. So set this room as a social distance conference room. There's 8 thermal mannequins were placed at the separate desks. Each was about 7 feet apart. We have particle sensors and CO2 sensors near the breathing zone of each mannequin. So on the left photo, we can see we have CO2 sensors and temperature sensors on each corner of the room, and we also have particle sensor and a CO2 sensor on the breathing zone of each mannequin. Next slide. For the mannequins, we have 8 thermal mannequins which provided about heat power about 100 watts to mimic human thermal plume. We have a laptop in front of each mannequin to mimic the work condition. So we have HVAC supplied via 4 registers on the ceiling. We have return via opening plenum above the ceiling. We release CO2 at the tracer for smaller aerosols. We also release 2 to 10 micrometer aerosols in many experiments. We test the HVAC in 4 thermal conditions, that is with HVAC off, with cool, neutral, and heated supply air. Next slide, please. The second setup is a simulated classroom with barrier. So it's almost the same settings as the conference room, but the mannequins was was located in 3 rows to mimic students, and there's 1 mannequin in front of the room to mimic the teacher. In some experiment, we put a barrier in front of the teacher, and we also release the contaminants from 3 different locations. 1 is from the teacher, and 1 is from the student at the first row, and some experiment was released from a student at the last row. Next slide, please. There are different mechanisms within the respiratory tract leading to the formation and emission of particles at different sites during these inhalation activities. Many studies have presented size distribution for exhalated aerosol. Inner study variabilities can be very large and is likely due to the difference in the sampling methods and the measurement devices. So in our studies, we most focus on the B mode and L mode, which is showing on the figure here. For the B mode, the small droplets expelled during the normal breathing. It's usually from 0.5 microns in diameter to about 5 microns. And the L mode is the energetic activity to vocal fold vibrations at the larynx. So it's likely to be like less than 1 microns to about 20 microns in diameter. For the O model, there are much larger droplets. It's produced between the lips and the epiglottis when the saliva is present. So it's larger droplets. It's usually like larger than 20 microns to about 1,000 microns. So in our study, we both focus on the B mode and L mode to mimic breathing and like normal speaking. Next slide, please. Since the concentration of exhaled aerosols from a single person are too small to be measured, so we have to simulate those emissions. Instead, using a real person, we generate particles and release them into the room at elevated concentration and track their movement around the indoor environment. We have 2 methods for generating particles continuously. For the 2 micron particles that are relevant for the B and L mode, we use a collision nebulizer. For the larger particles that capture the tail end of the L mode to mimic speaking, we produce liquid particles with TSI flow forecasting monodisperse aerosol generator, which we call the FMAC. Next slide, please. There are 3 release configurations in our experiment. In some experiments, we release smaller particles, like 2 micron particles with air and CO2. In some experiments, there's no particles, just air and CO2. And in some experiments, we release larger particles with air and CO2. I should mention here, all the release was heated to human body temperature, that is 37 Celsius, so to mimic real human inhalation activities. Next slide. So our experimental objective is to track the transport and fate of the particles around the indoor room, so which requires a lot of sensors to capture the spatial variation in the room in real time. So this instrumentation will include some 3D velocity probes, and we have some low-cost T30 CO2 sensors to measure the CO2 concentration, and we also have optical particle counters. We have a mini wide-range aerosol spectrometer in the middle of the room to tracking the size distribution of the aerosols we released. Next slide. Here is the example of 2 sets of experiments. So the release was from the mannequins at the bottom middle of the room, and the red line is the concentration measured on the mannequins on the right hand of the release mannequin, and the gray line was the medium concentration across the sensors in the room. So there's 2 releases in this experiment. The first release, the supply air is warmer than the room air. The second release, the supply air is cooler than the room air. For a typical experiment, we firstly usually flush the room for lower CO2 and particle concentration as a baseline. and then we could release particle and CO2 continuously for about 30 minutes, and then we stop release and let the HVAC system recirculate in the room air without a filter for 30 minutes, and then we turn on the portable air cleaner for 30 minutes to flush the room, and we will repeat the release and the flush period. From the two examples showing here, in the warmer release, you can see after the release period, in the decay period, the concentration of both CO2 and particle measured on the mannequins was larger than the medium level of across the room, indicating an imperfect mixing across the room. But for the cooler release in the second experiment, after the release in the decay period, the measured concentration in the mannequin was almost similar to the medium level, indicating a perfect mixing in the room. Next one, please. We found out the overhead heat produced stratified conditions. It's not showing here the figure. Oh, I was, yeah. Yes. Sure, here. So, and the cooling neutral airflows makes effectively to reduce exposures. On the figure on the left side, the y-axis is the height of the room, and the x-axis is the ratio of measured concentration at each location to the theoretical perfect mixing concentration, where we call it ERM, which is the ratio of observed to the perfect well-mixed, theoretical perfect mixed concentration. So from the first set of plot, we see when no AHU when the air handler is off, the thermal plume to drive the mixing. So there's some variability around the room. The room is not very well mixed. When the supply air was cool and neutral, the observed concentration is the ratio of observed concentration to the well mixed concentration is close to 1. Means there's a perfect mixing in the room. Also, mixing into the plenum above the ceiling reduce the concentration below those for well mixed room. Next one. Next, sorry, one more. No, previous one. Yeah. With heating supply air, the expelled respiratory airflows were concentrated in the breathing zone. This is a similar plot with neutral and heated supply air. And the X-ray is also the ratio of measured CO2 concentration to the theoretical concentration. So for the heated air, we saw the air supply remain near the ceiling at a very low concentration at high level, but at the mannequin level, the expelled respiratory flows accumulated in the breathing zone, which caused about 1.8 times compared to the well-mixed condition, and as much as 6 times compared to the well-mixed condition. Next slide. To break through the stratification condition, we operate 2 portable air cleaners. As shown on the orange plot in this figure, same as heated supply air, when running with the 2 portable air cleaners, so the air cleaners effectively make the room for lower exposures. So they reduce the exposure at the mannequin level from about 2 times to the well-mixed condition to almost 1, that's equal to the well-mixed condition. As mentioned here, the portable air cleaners also directly remove the particles from the filtration process. The next one. And we also conducted experiments with 20% auto air in the supply air to investigate the ventilation effect on dilution these airborne contaminants. So on the left side, the figures also shows the ratio of measured CO2 to theoretical concentration at neutral at classroom settings and also at conference settings with neutral air and heated air. So with the classroom settings, we saw that the imperfect mixing is overall the room, no matter what's the supply air conditions. So in the heated concentration, the stratification caused at least 25% of the mannequins to have exposure 2.5 times compared to perfect mixing. When it is cooled when it is cooling in the classroom at classroom condition, there's also imperfect mixing even with cool and neutral supply air, and the ratio of measured concentration to the theoretical well-mixed concentration is still between like 1 and 2 with 20% air. So that indicates that with poor mixing, this will cause the observed exposure all higher than the well-mixed concentration. So that will be a decrease effectiveness on the ventilation when imperfect mixing was presented in the room. Next one, please. Here is also an example of the spatial variations of our experiment. The figure here presents the baseline subtracted time resolved concentration around the room during the following release concentrations. So the conditions were no forced air, neutral supply air, cooling, and heating. Release was about 20 minutes, and the condition was maintained for another 20 minutes. So the released mannequins was on the top middle point, and the return was on the bottom right side. There's a air distributor, two air supply air distributors on the top left and the bottom right of the room. So with cooling and neutral airflow, the tracer quickly makes there's no hot spots, as seen as the black line with the black, blue, and the green line in the figure. And with the heated air, the stratification subsequently caused higher exposure all the location on the right side of the room, which showing on the red line, means all the exposure on the right side of the room was all higher by the stratification because the return on the right side. Next slide, please. So the conclusion of the study is the overhead HVAC system, which are common in schools and office buildings, can create a thermal stratified condition during heating, and the stratification interferes with dilution and reduce the effectiveness of ventilation to remove expel respiratory aerosols and can carry infectious agents. And sealing diffusers and achieve good mixing for effective dilution and ventilation of biobuffers, which supply cool or supply neutral air. And including portable air filters can break up stratification and improve the ventilation performance during the heating season. Next slide. In our future work, in our future work, we will study more larger particles release and their dispersion in the room and the different airflow conditions. Next slide. We have our paper published on indoor air, and if feel free to ask me any questions about that. Thank you so much. That's it.
Thank you so much for that presentation. We did have a few questions come in. If you have any questions, please use the questions function on your right control panel. The first one is, how did you decide to test at a 7-foot distance? Were closer distances considered in order to represent scenarios where people may need to sit closer together?
Yeah, we want to investigate how the aerosols was dispersed in the room with social distance required by the CDC. That's the objective of our design experiment.
Thank you. Another question, has any simulation been done in work settings such as offices with high occupancy with cubicle walls?
I haven't noticed about that studies, but we also have a team doing CFD simulations using the results of our experiment measured by the 3D velocity sensors to simulate some like various dispersion in occupied classroom and office room, which was conducted by the colleagues from University of Colorado.
Thank you. A few more questions coming in. 1 minute. Sorry about that. Have the findings of these studies with mannequins been verified with human experiments?
For human experiment, we have thermal mannequins which to mimic the thermal plume of the human. But for real human experiments, there will be breathing zone, right? Expel aerosols like at each location, at each mannequin. Mannequins do not breathe, right? Real human breathing their aerosols, which will in interaction with the released particles at the release point. Also, it's very hard to detecting the real aerosols expelled from the real human. The concentration are too small. So we release particles at a very high concentration to trace the dispersion of these aerosols. With the real human experiments, the expel flow was maybe really small, and the concentration, the detected concentration will be low, but the inhalation flow and exhalation flow from each human beings will also influence the airflow distribution across the room. That's the difference with using mannequins.
Okay, looking one more time to see if we have any further questions. All right, it doesn't look like it. Back to you, Lorene, to close us out. Thank you again.
Thanks.
Thank you. I want to thank both of our LBNL experts who have given us a lot to think about. And I'm looking forward to hearing more from this type of research in ways that we can translate that into engineering mechanisms or just human interactive mechanisms that would allow us to control not just SARS, but all kinds of viral transmissions. It really makes me wish I worked in labs where I could play with mannequins and have sneeze machines and that kind of stuff. But it was really, really interesting, and I do look forward to hearing much more from both of you. At this time, we hope that you found the information provided in this meeting helpful, and thank you all for participating. I want to remind you that the minutes and other materials from this meeting will be available on the CIAQ website within a few weeks. There will also be recordings posted of all of our speakers. The school's website you can also get to it from our EPA website and find that information that is application type information for work with our schools The next CIAQ meeting is currently scheduled for October. Is scheduled for February, and we hope you will join us then. And if there's nothing else from Lindsay or Christina, we will call the meeting and adjourn.